Now I'd like to introduce Ben Collen, who's been heading up the Biodiversity Indicator group at the Institute of Zoology London. We're grateful he made the trip over to here. He'll tell us about the work at the Institute as well as some work he's involved in with the Red List and ICN. So thanks a lot, Ben.
Thank you, Walter. Thanks for inviting me. And good afternoon, everyone. So the title of my talk is Tracking Global Biodiversity Change in Space and Time. And this is me actually doing that in the flesh. I'm going to talk about a lot of the sort of global aggregations of data sets, much like a couple of our previous speakers has. But this is me actually doing it in the field as well, so I'm trying to maintain a little bit of that as well. So biodiversity change and conservation decision-making is sort of a broad concept of what I want to talk about. I'm going to try and address three main questions. Firstly, how should we track the fate of species and populations? I'll talk to you about a couple of ways in which we've been doing that. Secondly, how can we predict their change in a way that is most useful? We've spent the past 6 or 7 years developing indicators for the Convention on Biological Diversity, but how can we adapt those and use them into the future? And thirdly, conservation funding is very limited, so how do we best prioritize its use? So this is the sort of background for biological diversity, the conservation of biological diversity over the last decade or so. The convention that most nations in the world are signed up to, signatories to this, agreed in 2002 to this 2010 target, and that was to reduce their impact on biodiversity and to do that at a range of different scales from the global to local level. These are the measures that were put forward to address whether or not that target had been met. On this side, you have measures of state, so the aspects of biodiversity that we're somewhat concerned about. In the middle, pressures on biodiversity, all of which are going up. Here are responses, things like protected areas, biodiversity aid, and here are the benefits that humans derive in some way from biodiversity. And in every way that we measured that, the 2010 target was not met. Those targets were sort of shaped into what have become known as the Aichi targets, many of which are for 2020. And they've been subdivided into 20 sub-targets, addressing much the same in general terms, but more focused, if you like, addressing underlying causes of biodiversity loss, reducing direct pressures on biodiversity, and so forth. So two measures that we use to measure biodiversity, the first of which is the ICN Red List. Red listing's been going on for 50 plus years. The organisation that I work for, Zoological Society of London, has been involved in that since the outset. And one of our former directors, Georgina Mace, is really the architect of the type of criteria, the tools they use to make these decisions over time. I'm going to assume that most people have heard of these, but very quickly there are quantitative criteria by which species can be put into these categories, from sort of safe as houses to over the edge, and at every grade in between that. At the bottom here, data deficient species, those where we don't have sufficient information to put them into those categories. And there's been a large body of scientists that have worked over time to produce assessments of individual species or groups of species. Here's the status of mammals, and it gives you information like 25% of mammals are threatened with extinction. We can see how that varies. We can see how that differs between systems. We can see where those species are. And representation on of species on the Red List has been growing rather quickly over recent decades. But really, how good is it? If we're trying to use this information to answer questions about biodiversity in general, how good is it? And the answer that we sort of decided on in about 2005 was that it actually wasn't very good. In red here in the bars of sort of these very broad groups of animals and plants are the species that are assessed, versus in the black those that haven't been assessed. And so this we saw as a real problem. There are a number of different ways in which you can tackle this. The process that we decided on was to try and implement a sampled approach to red listing. So here's my analogy. This is biodiversity, as with an iceberg. Much of it sits below the water. and you don't see it, we don't really know what's going on. So these are the non-assessed parts of biodiversity. Above the waterline, we've got those that we know about, principally birds, mammals, and amphibians. But if we can core down through that, if we can take a sample of the non-assessed as well as the assessed, then we can better explain what's happening with biodiversity and hopefully make better decisions. Just to say quickly, the ways in which you can work out that sample size is you can just draw from sampling theory. I hate to mention it at the moment, but this is exactly the same as polling organisations do when predicting an election, and we actually come up with very similar numbers to them in terms of how many people you have to ask before you can get the right answer. This is a subsample of the bird data, and it shows you that once you're at 1,000 species assessed at random from that group, you can tell what's going on in the whole group. And so this graph sort of wraps up what's about 5 or 6 years' work and the input of around about 1,000 scientists. But we've added many more groups to this sort of set of species that we can infer extinction risk about. And there's a few different interesting patterns. Firstly, we can see, and this was a great surprise to me, that of the invertebrate groups that we've assessed, broadly they show a very similar level of extinction risk to the more sort of well-known groups of birds, mammals, and amphibians. Similar again with plants. Much more variation in that extinction risk. So these grey bars here are those data-deficient species I talked a little bit about. So as you would expect, a sort of a larger number of those in there, but in general, similar levels of threat, 1 in 5 to 1 in 4 species threatened with extinction. And with that, we can really start to address what's happening. We can start to track the changes in categories, how those species fare over time. So here's a sort of famous example. In the year 2000, the polar bear was assessed as least concern. In 2007, it was upgraded to a category of vulnerable on account of shrinkage in its primary habitat. If we do that with lots of groups, lots of species across groups, then you can get the change in categories from sort of better to worse. So a couple of things to note on this graph. Firstly, the rate at which these different groups are changing. So corals there versus amphibians here. And the second thing to note is their position on the y-axis. So the further down here you are, the more threatened species you have in that group. We can also start to pull together information about the threatening processes that are causing those species to fall into those categories of extinction. So this is an example of what threats and controls of threats have led to changes in categories in birds and mammals. So in red, you've got deteriorating species. In green, you've got improving species. Mammals in the solid bars, birds in the hatched bars. A couple of kind of interesting things pop out there. Most birds that are improving in status are doing so because of control in alien invasive species. Most birds that are getting worse, that are deteriorating in status, are doing so because of agricultural practice. So there's a mismatch there between what's causing the threats and what we're doing about those threats. And that's all useful information at a global level. One of the really important pieces of information that any Red List assessment gives you is a geographic range map for that species. So here's the African elephant, and the range map for those species. And there's been a real kind of mini industry in pulling these together. You can aggregate those maps over a global surface. So here is a mammal richness map of the world. But as we've introduced those new groups, as we've started to understand how they're distributed, we can see that some of the patterns that they cause are very different. So this map here is of freshwater crabs and freshwater crayfish across the globe. And you can see two groups of species in a very similar ecological niche, shredder communities in particular, really subdividing the globe. A couple of other examples, a marine one, reef-building corals here. You can see where the sort of centres of richness for those are versus lobsters, if we can move on, where they're really rather different centres. And that matters for conservation planning. Obviously, if you're going to put resources into an area, you need to know what species are there and what's important. If we look at congruence, between two different measures. So on the left-hand side here of richness, and on the right-hand side of threatened richness in a sort of a matrix, you can see some really low numbers there is the thing to take away from this figure. So congruence across freshwater taxa, which are increasingly higher levels of threat than those in other systems, are really rather poor. And what that means is that if we're going to better design conservation strategies, we need to assess more species and have a much fuller picture of how biodiversity is changing. So here's just one example. The lower panel are the top 5% richest cells of threatened birds, mammals, amphibians, and on the top, same measure, but for freshwater fish, dragonflies, and damselflies, just because they're the three first groups that we did. And you can see some startlingly large areas of the world where if you focused if you took the information from the top set, you would choose as the areas to concentrate on. If you took your information from the bottom set, you'd leave them alone. So conclusions for that sort of particular section. Coverage of risk metrics and range data are continually improving across taxa. Congruence across taxa at a global scale at least are very varied, and that makes the task of conservation planning much more difficult. With enhanced spatial data sets and better taxonomic coverage, that will allow us to better design things like protected area systems, and the marine system really kind of lags behind some of the terrestrial data sets in the main, but there are people that are working on that, so hopefully that'll pick up in the next few years. Second important measure of biodiversity change is that of change in abundance. And here I'm going to talk about the Living Planet Index. Living Planet Index was a kind of a state of the environment report developed by WWF back in 1997, I think first launched in 99 or 2000. We became involved with them in 2006 in order to try and strengthen the data behind this report and do some new things with it. And what we do is we collect abundance data across vertebrate populations. Various different sources and types of data. The majority of them are from peer-reviewed primary journal literature, but also NGO literature, the grey literature, national parks and protected area networks, and so forth. Different measures for different species. We're talking about all vertebrates here, so you can't be really kind of dogmatic about the way in which data are collected. So we can get things like total population estimates, obviously the best thing that we can get, density measures, biomass estimates, things that relate to or correlate with abundance, like number of nests for marine turtles. And then we have a number of different criteria against any given publication. It has to be traceable, it has to be measured over time with the same method. We have to know where that was done and who did it and so forth. So a few examples of the types of data that you might collect. So this species, southern right whale from Indian Ocean, showing a gradual increase in population number. Wandering albatross from Bird Island, South Georgia, showing a slow, steady decline. And the loggerhead turtle showing a population crash. So the data set's made up of just over almost 2,700 species now, roughly equally balanced across temperate and tropical zones, and that amounts to some 9,000 populations. I think we have about 13,000 in the data set as a whole now, 9,000 of which are used to calculate the index. It's all going online very soon, and what that shows us, sorry, a bit more about the data set. This is the distribution of all those abundance time series data across the globe. So, you know, some areas where we're good, some areas where we'd like to be better. And if you aggregate those data up, what they show you is that over a 35-year time period, we've lost around about a 30% on average, 30% of the abundance of wild vertebrate fauna. And that's a lot worse in tropical systems than it is in temperate systems, largely because temperate systems have probably gone through those changes rather earlier. And to set that against a backdrop, over that same time period, human population has practically doubled. So that's sort of the output of the report, the kind of the broad headlines, and WWF is superb at getting that message out. That report is produced in 13 different languages. The print run is in the tens of thousands, the downloads are in the hundreds of thousands. As well as improving that data set, we've been trying to focus it on new things as well. So here's just one example in the Arctic. So looking down on the top of the globe here, these are the abundance data sets that we've managed to collate for that region. And if you do the same thing, if you aggregate those data up, you get different patterns. And if you start to pull those patterns apart, you can start to discern what's happening in different regions. So here, high Arctic species over that time period have shown about a 20% decline in comparison to low Arctic species, which is, because of the geography of this area, primarily marine species, where control of hunting has led to increase in size. Overall aggregated abundance trends are actually very positive, up to a 50% increase over that same time period. And in the subarctic, sort of moving along, if you like, on a steady state since about the 70s. And then what we've looked to do is to try and look at whether we can predict why those changes have happened, in particular in a spatial context, what land use changes have gone on, what biology can explain those changes. Can we pull out some sort of broad measures to describe what's going on? And the short answer is it's really quite difficult, and we've been unsuccessful in really pinning down exactly what's going on. So this map here shows you the distribution of those abundance time series again, in the sort of the hotter colors, those time series that are declining quite rapidly, in the cool colors, those that are increasing. And we evaluated a range of different sort of spatial measures of change: temperature range, anthropogenic land cover, natural land cover change, protection, human population density, all of those things. We were only able to explain about 10 to 15% of the variance in those data sets. And this is a relatively recent piece of work, so we're going on to sort of trial some new methods and look for some better explanatory variables for that sort of pattern. But that's where we're really starting to take these data sets now. So through our involvement with those types of biodiversity indicators, we came to realize that they were being used as tools for embarrassment. You know, you could walk up to a set of policymakers and say, look, you know, biodiversity's declined by 30% and it happened on your watch. And they say, what should we do about it? And we didn't have a good answer. So now we're trying to get into sort of a more predictive framework, in particular to try and inform and influence policy. So the current set of indicators selected by the CBD are really poorly targeted at solutions. So we wanted to know, can we do better? And what we decided to do was develop a framework to test different policy options. And we drew out a couple of case studies to do that, which I'll tell you about. And really this graph explains what we're trying to do. This is your index of change, be it abundance or risk, or it might be range size, it could be anything you like. What we'd really like to be able to do is give policymakers these options. You know, this is policy option A, B, and C. This is what we think is going to happen under those set of circumstances. This is how much it's going to cost to do those things, and really start to give people tools to make decisions with. For our first sort of attempt at this, we targeted CBD Strategic Goal C, which is about protected areas. Here's the goal: by 2020, at least 17% of terrestrial and inland water should be under protection. In effect, 17% of the globe should be under protected areas. It's a politically derived decision rather than one that's got a huge amount of scientific basis. We took all our information from African protected areas. This was published by a PhD student of mine, Ian Craigie, back in 2011. And what it shows is in those protected areas across Africa, a 50% decline in abundance. Regionally, it shows rather stark differences. So Eastern, and particularly Western Africa, are faring much worse than Southern Africa, where a greater history of protection is in place. So we took that population data set, we took a spatial data set of if we were going to expand protected areas, where should that happen for the sort of in a systematic way for the best benefit of species. We drew together estimates of populations in 41 countries, 53 species of mammals, and we, in effect, grew those populations forward according to the regional trends that we'd observed in the recent past. We then reassessed every decade what those data told us, calculated abundance trends, so the Living Planet Index, calculated total abundance trends, and we calculated the Red List Index as well. So really this is getting at a couple of things. Firstly, what's happening. Secondly, can our indicators pick up those changes? And we used 4 different scenarios. One was business as usual, just keep going as we are. Second, expand protected areas to 17%. Thirdly, make protected areas effective. We've seen that they're not effective. And 4, do both. And we made a bunch of assumptions. For brevity, I'll forego those, but I'm happy to talk about them afterwards. And this is what we showed. So this is the Red List Index that I showed you earlier on, but just for these African mammals in protected areas. And it shows you that without effective management, protective area expansion will be of little value. And that shows us really a couple of things. Firstly, that perhaps our target is misguided if we believe these results. It shouldn't be about expansion. It should actually be about the things which enhance protection. And secondly, it shows us that one of our indicators at least can pick up those trends. So that's a positive thing for that indicator. A little more detail. This is the Living Planet Index. So that measure of abundance rather than risk, showing much the same story, certainly the same trajectories. But a couple of key differences between the indicators. So I've just pulled out this one species just to sort of exemplify. This is what happens under one of the scenarios for the abundance of that species across those regions. So that's if you can track total abundance, which let's remind ourselves that most monitoring programs can't, but that's your best case data set. If you create an index of those using abundance, you keep track of the fact that this has dropped away to nothing, or close to nothing. If you use a Red List Index, so tracking the extinction risk of that species, you get this really counterintuitive effect whereby down here, the species gets assessed as least concern again, because it's in protected areas, fences around them, only in southern Africa. It's been extirpated from the rest of its range, but in effect, this index forgets that historical baseline. And that's an unintended consequence that we didn't realize when we were sort of putting this indicator together. So a couple of conclusions. Expansion is futile without effective management. Target achievement can therefore be misleading if we don't measure the right things. Both of our indicators seem to be able to measure those trends, but perhaps there were some unintended consequences. And the main caveat of this is it was a very simple sort of proof of concept trend-based response. There was no process involved, and that would be a way to develop this in the future. So finally, I want to go a little bit into prioritizing conservation under limited resources, just in the last sort of 5 minutes. Talked about how we pull together all those different types of data on risk and abundance, but how do we actually employ those? How do we use those to make conservation decisions? It's been framed as a sort of the agony of choice. There are many deserving causes out there. Where do you choose to put your conservation buck? And there are lots of ideas on how to do this: threatened species, restricted range endemics, flagship species, indicator species, those of economic value, and so forth. But an idea that came really from the literature in the early 90s was this idea that phylogenetic diversity or evolutionary history, I'll use the terms interchangeably, really give you something that those other measures perhaps don't. And what we should really be doing is conserving evolutionary potential of phylogenetic diversity. Because that's a compound metric of lots of different things that we value: genotypic diversity, character, feature, ecological diversity, and so forth. So maximizing PD not only gives you the most of what you've got, but it gives you the most options for the future. So a little cartoon example of that, made-up phylogeny. Here's the echidna out on a branch on its own. If you had to choose which of species A, B, or C went extinct, You probably wouldn't choose the echidna because that would lose you 56 million years of independent evolutionary history, but rather you choose one of these species of rodent, which would lose you a few million. You also know that there's another species which is very like it, which is going to come back and take its place. So that's the concept. But that had never been implemented in a conservation program, really for kind of three fundamental reasons that I can see. Firstly, branching patterns and lengths of branches is a difficult thing to do. It's really only with the advent of molecular phylogeny that we've been able to bring together the tools to be able to implement this type of approach. Secondly, it's a little bit nerdy. It's quite difficult to convince the public that phylogenetic diversity is something that they should be concerned about. And perhaps that's the reason why it hadn't been attempted. And finally, there's been a lot of investment in alternative approaches. So really, why rock the boat? We decided to anyway. And so what you require here is a super tree giving you the relationship of all mammals. That's where we are. And combine that with a measure of use that to calculate evolutionary diversity or distinctiveness, phylogenetic distinctiveness, which is in effect the length of any branch over the number of species that subtend it. Again, for brevity, I'll breeze through some of the methods, but one of the things to deal with is polytomies, where you don't have complete information about any branch, any given branch length or structure. We had a couple of ways to deal with that, which we can go through. And we estimated the variance around that. Take your measure of urgency from the red list, so just read those categories as read because they relate directly to extinction risk, and combine that into something that's mathematically sensible. So each increment in category is a doubling of extinction risk. So this just combines those two measures together. But what this really is, although this is a mathematically sensible way to do it, what we've in effect done is taken two distributions of phylogenetic diversity and endangerment, and we've said, well, if we had to split this up, this is the one we'd might like most, and then we'd like these species second, and these ones third, so forth. So there's an element of sort of an arbitrary decision-making about this as well. A couple of the facets of this, you get a good broad distribution of different threat types if you use this. If you use this method, so critically endangered, endangered, and vulnerable species all in the top 100, we can look at how robust it is to change. I'll flick through this one. We can look at how. Oh, that's loud now. We can look at how performance changes as the red list changes. So we can compare genuine changes to non-genuine changes. We can look at how red list data, how things move between recategorizations. So data deficient species can move into any category. Threatened species tend to stay threatened, and species can become unextinct, if you like. So we need to understand that those are okay in our ranking system. We can look at the impacts of taxonomy, whether we split or lump things, whether that affects that ranking list, that list of species we think are most important. And we can look at the change in ranks over time. So to wrap this up, we sort of put our money where our mouth is and went out and, having developed this academic approach, we went and implemented it. You can go to the website, zsl.org/edge. It's a very rich website, lots of blogs attaching people to the projects in the field. We focused on those species where no conservation attention was being placed. It gives you your focal species. These are some of them for the mammals. We have a sort of a fellowship program with in-country scientists. We work with them over a 2 to 3 year period, and they actually implement the science behind this method and the science behind the conservation program that we're trying to implement. We've expanded out over a number of groups: corals, amphibians, birds are on the way, and we sold it to the public. Our press officers, when we told them about the idea, told us just to not bother. We were doing interviews for 2 weeks solidly when we launched this in 2007. So I don't know whether it's something about the UK, whether it's something about sort of rooting for the underdog, but it certainly struck a chord. And there are some signs of success. We get 1 million, we got over 1 million unique website visits on day one, which is now 50,000 unique visits a month. We've got a community of scientists involved. We've funded 50 different conservation fellows around the world on 2-year schemes. But the real kind of test of this really will be, do the species improve in status? And that's something that only time will tell. Those are my acknowledgements. A lot of people have been involved in different aspects of this work, and these are funders and data partners. Thank you all for your attention, and I'm happy to take any questions.
Thanks a lot, Ben. We're a little over time here. So, Gerardo, why don't you already come up and get started here? But let's grab a couple of questions at least. So that's Woody over there.
Yeah. Ben, has anybody worked on combining the edge species with the Alliance for Zero Extinction, the AZE sites, to sort of put in place and monitoring element to the edge species?
Not that I know of. We started plotting out the spatial distribution of those species and looked at how the phylogenetic measures correlate with those. I don't think anyone's looking at these. We'll be able to get
a monitoring layer over the top.
Yeah.
Another question? That's great, because then we can move on to Gerardo. Again, Ben is around. Thanks a lot, Ben.